Okay, next gene mutation we're going to look at is insertion. Okay, and we're going to look at insertion and then we'll do a separate video for deletion. And these are classed as frame shift mutations, okay, because as we said before in the previous video, that every amino acid after, so every codon, which therefore gives you a code for a particular specific amino acid, is therefore affected after where that mutation is, okay? So as you see here, this is what we would normally expect. So here is our specific code for these series of amino acids, which is therefore going to create a protein for us, okay? So here is our nucleotides within our DNA, okay? And our messenger RNA, okay, so our complementary pairing, so C and G, A and T, blah, blah, so on and so forth, okay? So as we know, messenger RNA, 3, so a triplet of bases, so our 3 nucleotides, therefore gives us our codons, okay? And in a perfect world, these codons, okay, therefore gives us these 3 types of amino acids, okay? Remember, amino acids attached to a transfer RNA. So we're going to get glutamate, glutamate and glutamate. This is our 3 amino acids that we are expecting, okay? However, with our insertion mutation, what's going to happen is, so if we look at our normal or expected code for the protein we're looking for, it would be C, T, C, C, T, C. Okay, so CTC and then CTC, CTC, CTC. But what we notice with this one is there's an insertion, so there is an extra nucleotide inserted into this sequence. So because the cell has to still go through the process of recognising in order the sequence of nucleotides, we therefore get a completely different amino acid. So G, A, G, so remember this is our messenger RNA, we get 1 codon. But this inserted nucleotide, we therefore have CCT, which therefore gives us a completely different codon from the last one, from what we'd expect. We'd expect this to be GAG, and we now have GGA. So again, giving us a different codon, which therefore means that every other sequence after this is going to be changed. Okay, which therefore gives us GGA and then GG. Okay, so as you can see, the amino acid that's actually produced, so we've got our glutamate here, but because of this inserted nucleotide, we therefore have glycine as the second amino acid and glycine as the third amino acid. Okay, so our insertion mutation, okay, is classed as a frame shift mutation. Okay, so 1 nucleotide is added and the entire sequence is wrong from then on. So all amino acids are altered from then on. And what we tend to say is with insertion mutations and deletion mutations, they end up technically being fatal. So they can have catastrophic effects actually on the organism as such. Okay, so insertion mutations.